So welcome everyone. Today's tutorial will be on Mendelian randomization, focusing on an introduction to the area of Mendelian randomization. My name is David Ryan. I'm a junior doctor and researcher based at St. George's University in London. And Mendelian randomization is quite a really fascinating paradigm that uses genetic data to explore the relationship between exposures, such as drug exposures, on outcomes, and increasingly being used in drug discovery pipelines to identify new drug targets and to kind of delineate potential side effects of new drugs and existing drugs as well. So I think it's a really exciting area, and there's a lot of really interesting research in this area as well. The plan for today's session, 5 key aims. Firstly, I'm going to do a little bit of a revision of study design and research methodology, focusing then on the limitations of observational studies and a little bit of a primer on genome-wide association studies as well. Then we'll discuss a little bit about what Mendelian randomization is and how we set up this research methods. And then we'll give a little bit of a walkthrough of how we go about a simple Mendelian randomization study as an example to kind of bring the concept to light and make it more concrete. But firstly, we just revised existing study design and methodology. So really, study designs can be split into 3. So the top you have interventional studies, and this would be a randomized controlled trial, and these are seen really as one of the best methods to explore causality. So, for example, recently there's been lots of randomized controlled trials using looking at new drugs and therapeutics for COVID-19. You've had the major vaccine studies. So these are really kind of almost seen as a gold standard to look at whether a drug is effective or not. However, above that, then you have summary studies which combine many different randomized controlled trials, and this increases the power and statistical power. So systematic review is when you systematically go through literature and try and gather the conclusions from different randomized controlled trials to see, well, what evidence is there for a certain treatment, for example. Then in meta-analysis, you actually perform a statistical analysis where you pool the results of multiple randomized controlled trials together to get what we call a pooled estimate, and that is seen as a more accurate representation of the true state of affairs. So again, these are really quite powerful studies because they look at many, many different randomized controlled trials. And then you also have observational studies, and these don't make any intervention, so these kind of observe what is happening in natural phenomena without making any intervention, and these include case control, cohort, and cross-sectional studies. And we can arrange these studies in a pyramid. There's a hierarchy of study designs with systematic review and meta-analysis providing the best evidence to support or refute a treatment. And again, the top being the most valid form of study design when it comes to evidence-based medicine. Then underneath that, we would have a single good quality randomized controlled trial. And then below that, we have observational studies, because the evidence we get from observational studies is seen as weaker because there's a number of issues with these studies that we will go on to discuss. But as I said, randomized controlled trial is one of the most is seen as really one of the gold standards to assess whether a treatment is effective or not. And here I have the basic structure. So we take a sample out of the population and we randomize people to either receive the treatment or else another treatment, or that could also be a controlled, like a placebo if it was placebo controlled, placebo being there was no active therapeutic ingredient in that intervention arm. And then we follow these people up to see, well, does the different treatments have any change on their outcome? And really the benefit of a randomized controlled trial lies in the randomization. This makes sure that the people who receive treatment A or treatment B are similar in terms of their baseline characteristics. for example, that there are similar distribution of ages in both the treatment A and treatment B, that there's similar proportion of people who have different comorbidities such as diabetes. So really, because these different treatment groups and the people who receive treatment A and treatment B are similar, the only difference between them is their allocation to receive either treatment A or treatment B. We can then, when we look at the outcomes, say that, well, any difference in outcome can only be attributed to the different treatments that they received. So again, the only difference between treatment A and treatment B is their allocation. Everything else, all the other demographic differences, differences in ethnicity, everything else is balanced. The only difference is the treatment allocation. I'm going to introduce a new term here, which is confounding. And for example, all those baseline differences, demographic differences, comorbidity differences that could exist between people, these are called confounding factors. And a confounder is a variable that is associated with both factors, and this creates spurious associations. So I'm going to explain that in a little bit more detail because it's quite a key concept. Confounding factor, as I said, associates with both the exposure and the outcome. So if I was to do a simple observation, we could see that as the sales of ice cream increase, there is an increase in shark attacks. And one could say that, well, actually, does that mean that ice cream sales are causing an increase in shark attacks? And we know that is really unlikely to be true and quite spurious and fantastical. So there could be an alternative explanation. And here we have the alternative explanation being the confounding factor. And that's because as, you know, if it's sunny, people go to the beach, they tend to buy ice cream. But also as people go to the beach and it's sunny weather, they also go swimming and shark attacks increase. So we have a common cause for both ice cream sales and shark attacks. That's the beach weather. And this is creating the spurious association. So the confounding factor is actually what's causing the appearance of there being an association between ice cream sales and shark attacks, but it means that ice cream sales do not cause shark attacks. So then, to give an example of how a study would be if there wasn't any randomization and how confounding comes into play, we could look at a non-randomized controlled trial. So let's say a major pharmaceutical company wanted to set up a new COVID-19 vaccine. However, instead of randomizing people kind of properly, they decided to allocate people to receive either vaccine A or vaccine B according to where they are. So they gave the vaccine A to people in hospital and they gave the vaccine B to people who were in the community. And then they followed them up and saw, well, what proportion of each treatment arm had severe COVID-19. And as you would probably expect, people in the community who received vaccine B had less severe COVID-19. So therefore, the pharmaceutical company could conclude that vaccine B that was only used in the community was better than vaccine A in terms of severity of COVID-19. However, this is a spurious association because there's a confounding factor, and that's the fact that the people who were in hospital are very different to the people who are in the community. So for example, people in hospital would have higher rates of diabetes, would be more frail, so they'd have a lot more comorbidities. And these people only received vaccine A and they had a higher severity of COVID-19, but we could see that actually that association could be mediated by the increased prevalence of diabetes in that cohort. So actually the association is spurious, and we cannot say that treatment A was better than treatment B because the baseline differences between the allocated treatment groups was very, very different. However, if we did randomize this trial and did it more appropriately, and allocated people randomly to receive either treatment A or treatment B, we could then more appropriately infer whether one treatment was better than the other, because actually the only difference between these treatment groups was their treatment. So if there's a difference, we know that that must be due to the treatment itself. Therefore, we see that actually the randomization balancing confounding is really important to establish causation. Moving on to now some observational studies, and again, these are slightly below, below randomized controlled trials in terms of the hierarchy of evidence-based medicine. I won't delve too much into this as a whole area of study design, but just as an overview, case control studies, this might be used to look at the effect of a risk factor on an outcome. So we look at a population, and then we take a sample of that population, and we find people who would have the outcome of interest. So this could be, for example, lung cancer and people who didn't have lung cancer. So we establish who was a case and who was a control. And then we look back in time, so it's retrospective in nature, to see, well, what proportion of the patients who had lung cancer were exposed to something like asbestos. And given the proportion of people who were exposed to not exposed in the cases compared to the controls, we can work out the odds ratio for lung cancer given exposure to asbestos, for example. Similarly, a cohort study is another form of observational study, oftentimes used to explore the association between risk factors and exposures. But instead of starting with who actually had or didn't have the disease, we look at people who had the exposure and who didn't have the exposure. And then we assess, well, what was the likelihood that these people would then go on to actually have the disease of interest or the outcome of interest. Cohort studies can be prospective or retrospective, so we can follow people up over time in the future, which is called prospective, or we can look back, so look at who had an exposure and then who had subsequently then developed the outcome. So that's actually the key difference between cohort and case control, is what we actually start with. So case control, you start with who was a case and who was a control, and with cohort studies, you look at exposures at the start. But the major issues with observational studies, and the reason they're below the randomized controlled trial, systematic review, meta-analysis, is because of some key issues with observational studies, namely, most of them would be retrospective. So actually, this means that they could be open to bias. And what do I mean by bias? So this means that there's a systematic difference between patients who were exposed to the risk factor versus those who weren't exposed to the risk factor, for example. And this could be due to many different reasons, and we can't control for that because actually we're not making an intervention. So for example, if I was looking at the association between, for example, a drug like statin and risk of pneumonia, for example. And I was looking at this from an observation study. So this would be a cohort study where the exposure would be whether the patient was exposed to a statin. And then I followed them up to see how many of them had died from something like a pneumonia. But actually that relationship could be due to several other different, as I said, confounding factors, that people who are prescribed a statin would be older, they might have more comorbidities. So actually because we can't control these trials and there's a lot of other variables at play and they're not randomized, actually we can't infer causality because there's so many confounding factors. And also there's risk of bias. And there's another thing called reverse causality, which I'll go on to explain, which means that actually we can't infer causality from observational studies because we're not sure which direction the causal relationship is occurring in. So, as I said, reverse causality here is when the presumed association actually in reality is happening in reverse. So for an example, if I was doing a study looking at the relationship between cannabis use and schizophrenia, One might think that the presumed association would be that cannabis use causes schizophrenia. However, actually, this could also be happening in reverse. So it could be reverse causality, where patients with schizophrenia might be experiencing distressing symptoms and they might use cannabis as a way of treating and alleviating these distressing symptoms. So actually, we don't know whether cannabis is causing schizophrenia or whether schizophrenia could be causing people to use cannabis. So not only is there risk of confounding, there's also the risk that, well, actually the true association might be masked because it could be happening in reverse. So kind of to summarize the advantages and disadvantages of observational studies. First, the advantages, they're economically quite efficient because a lot of the data can be already be captured in databases that we can access quickly and with very little cost involved. And they can be done quickly, particularly if they're retrospective in nature and the exposure and the outcome has already taken place. However, as I said, the disadvantage is that we can't infer causality using observational studies alone. There's also, as I said, confounding, and this can be both measured and unmeasured. We can never fully capture all the infinite variables that could be confounding an observed association. And as I said, there could be reverse causality at play. There's also a lot of bias in observational studies that we need to be aware of. But I think the main take-home message for this part of the tutorial would be that association does not mean causation. Simply because we see that two variables are related doesn't mean that one is causing the other. It could be confounded association, it could be reverse causality. So we just always need to keep this in mind when we observe an association in nature. I'm now going to shift focus a little bit and have a quick discussion about genetics, and where this is probably quite a lot of revision for you, but because we'll be using a lot of genetic data, I just want to make sure that we have a clear understanding about some key terminology. So as we know, the central dogma of biology is that DNA encodes RNA, which then encodes a protein. DNA has the double helical structure, and its basic building blocks are nucleotides, so A, T, C, and G. And then this DNA encodes genes. So a segment of DNA would be called a gene, and this would control different traits. So for example, green eye color. And there can be many millions, billions of genes. And these all encode different physical traits or phenotypes. Allele is another term, and it refers to different versions of the same gene. And then a term we'll come across a lot in Mendelian randomization is the concept of a single nucleotide polymorphism, or a SNP, SNP. And this is when one nucleotide is replaced with another. So you could have an A replacing with a T, or a C being replaced with a G. So it's a different variant of the gene, and this can change how the gene functions, and it can be associated with risk for different diseases, for example. And this is really what we're looking at in terms of Mendelian randomization and the genetic variant, is a single nucleotide polymorphism. And the study we get our data from in Mendelian randomization is known as a Genomewide association study, or a GWAS. And this is an observational study which incorporates many, many individuals. And then the researchers search the genetic data for all these individuals and looks at, well, what genetic variants are associated with different traits. So for example, if I was doing a GWAS for type 2 diabetes, I would get a huge cohort of individuals, and I would get their genetic sequences and their genome. I would then look at people who had type 2 diabetes and didn't have type 2 diabetes, and I would see, well, what genetic variants were more prevalent and more associated with type 2 diabetes compared to the people who didn't. So again, I've taken the individuals, gotten their genetic variants, and I've gotten a phenotype or a trait that I want to look at, and then I look at the association between the different traits and the variants. And essentially, a genome-wide association study can be conceptualized as a really large regression analysis. So I'm going to go in a little bit more detail about regression analysis, but basically, if it's a binary trait that I'm looking at, so if it's a trait like type 2 diabetes, which is a yes or a no, there's only two outcomes possible. I have diabetes or I don't have diabetes. The regression we use is a type of regression called a logistic regression. And the output of this model is the log odds ratio of type 2 diabetes given a certain SNP. Whereas for continuous traits, and that might be something like my HbA1c, which is a glycated hemoglobin A, and measurement of kind of long-term glucose control, that would then be modeled using a linear regression model, and this shows the change in HbA1c associated with SNP. So again, it's genome-wide association study is a large-scale regression analysis that looks at the association between variants and traits. If the trait is binary, has either disease, yes or no, it's a logistic regression. If it's a continuous trait where the value of the outcome can take an array of different outcomes, so from like 0 to 100 or any other kind of continuous spectrum, then that is modeled using linear regression. So a little bit more detail about linear regression. It's a statistical technique that describes or models the relationship between an outcome and predictor variables. So it kind of goes back to some basic mathematical concepts where we fit a straight line equation to describe data. So the example here on the right-hand side is trying to predict body weight given a person's height, or a baby's height, for example. So on the y-axis, we have the outcome variable, and this by convention is called the dependent variable. Okay, and the x-axis is the height, and this is the independent variable. Okay, and this is a variable that we could substitute in the values into the equation to find out, well, what the predicted body weight is. So we've gotten these observations, and now we fit a line. So then if we want to take a different height, we could, let's say, plot here and go up to the equation and then come across and get the predicted body weight. So we're using data to build a model that then describes the relationship between the dependent variable and the independent variable, or the outcome and predictor variables. And again, so this is the equation we use. It's y is equal to mx plus c, where m is the slope. So that's the pitch of the line, and that's created by the angle between the line and the x-axis. And then we also take into account the intercept, and that is where the line crosses the y-axis. So again, this is a simple straight line equation to fit the data. And the purpose of this modeling technique, firstly, that we can predict an outcome given new values. So let's say if a baby's height was, let's say, 55, we could then go across and predict their body weight, given that we've established the model. But also quite an interesting concept is when we build this model, we can control for various factors. We can start to understand the effect of changing one variable while keeping the other variables constant. So now we've gone on and collected data about the baby's term at delivery. So were they early, were they late in terms of their gestational age? And then we created a new model, and that shows that weight is equal to 80, that's intercept, plus 2 times the baby's height, plus 5 times their term at delivery. So this can be interpreted as keeping, if we want to look at the effect of height on a baby's weight, controlling for their gestational age, or how old they were when they were delivered, we could say that for every increase in unit of height, so that's for every inch in height, their weight would increase by twofold. And that is keeping delivery at term constant. Okay, so we can see that we can control for variables using linear regression. So there's an increase of 2 for every unit increase in height. So this is really the purpose of regression model, is to build the model, and then we can understand the association between outcomes and predictors, but we can also control for other variables. So we discussed linear regression where the outcome is continuous. Now we're considering logistic regression when the outcome takes the form of 0 or 1. So it's a binary disease, but binary outcomes, so you either have the disease or you don't have the disease. However, we can't fit a straight line equation in this instance because it just doesn't make sense. When the outcome takes a form of 1, you have the disease, or 0, you don't have the disease. We can't really predict this using a straight line equation. So therefore, we fit a different type of equation. We fit we use a logit function to fit a sigmoid shape curve. Okay, and this shows essentially the new equation, and this bounds the output of the model between 0 and 1. And the importance of that is that this means that we can use this as almost like a probability, that the probability of having the outcomes of the dependent variable is 0 or 1. So that shows that it lies within the range of 0 and 1. And the outcome here from logistic regression is the log odds. Okay, and that's to do with the equation that we use as the sigmoid function. It gives you the log odds ratio of the outcome given the independent variables. And the odds ratio is simply the probability of the event divided by the probability of the event not occurring. So it shows us, well, how likely is the dependent variable given these number of independent variables, and it gives us an idea it gives us the odds ratio, which then is essentially talking about the likelihood of the event happening. Okay, so it's a little bit more complicated than logistic regression, but for binary traits, it's how we model the association between a binary outcome and the predictive variables. So applying these to a genome-wide association study, we're looking at type 2 diabetes, which is a binary trait. This then looks at, well, how is the association between all of these variants and the binary trait, and it shows us the odds. So what is a log odds for type 2 diabetes given a certain variant or a certain single nucleotide polymorphism? And for linear regression, it shows us, well, what is the change in the continuous trait, so something like HbA1c, given this variant, this genetic variant. So that's how we get the genome-wide association study data and what it outputs. And this is the data that we get from a genome-wide association study. And I've taken this from the MAGIC Consortium genome-wide association study for type 2 diabetes. And SNP here refers to the single nucleotide polymorphism, and that is the genetic variant. And we'll see here that all of them are start with ORS, and this is like the code that is unique for that SNP in question. So this refers to the fact that at chromosome 6, at this position, if you have the A nucleotide in this position, and because of this is type 2 diabetes, the beta here refers to the log odds. Okay, and that means that there is a slight increase in the log odds of, actually, I think these have been exponentiated into the odds ratio. So how do you convert log odds to the odds ratio? Is that you use exponential function Okay, so this means that there's a 1% increase in the odds of type 2 diabetes if you have this allele or this nucleoside at this position in this chromosome. However, you see that this is not significant. Okay, SE stands for the standard error, and that's a measurement of the accuracy for this estimate. However, we'll look at the bottom line here, and this shows that this SNP, which is T allele at chromosome 10 at this position, this is highly significant. Okay, so it's almost 5 by 10 to the minus 68 in terms of its p-value. There's a 37% increase in the odds ratio of type 2 diabetes if you have this genetic variant. Okay, and this is a summary. This is summary data from the Genome-Wide Association study. So I've only taken 10 examples from that study, but I think there's something like 2 million different SNPs. So it's quite a large file, and this is the outcome of that study. So this shows us the association between various genetic variants called SNPs, and the beta is the strength of that association. So it describes what is the odds ratio of type 2 diabetes given that genetic variant. Okay, there's a couple of more slides just about genome-wide association studies, and then we're going to have a quick break after that because I appreciate this is quite a lot of information. But again, the beta is the regression coefficient, so that is the odds ratio for a binary trait, or the change in the outcome for every copy of the allele a person has. So for example, for each of, because we have two alleles, for each allele for each of these SNPs, if you have one of these SNPs in your genome, you have a 1% increase in the odds ratio for type 2 diabetes. However, if you have two, then it's per that allele increase, per change in the independent variable, and that here is the SNP. We get an increase in the odds ratio for the outcome. And that's called the beta, refers to the regression coefficient. The standard error, as I said, it was an accuracy measurement for the association estimate, and that can be used to calculate confidence intervals and the p-value. So confidence interval commonly would be the 95% confidence interval, and this is a range of values that we have a 95% probability that the true value occurs within this range, and that's calculated using the standard error of the estimate. So we look at the estimate and then we define the upper and the lower boundary based on multiplying the estimate, either by adding or subtracting. That should be a 1.96 times the standard error of the estimate, and this is based on kind of normal distributions, and so really it just calculates the 95% range, a range of values, but we have a 95% probability that the true association or the true estimate lies within. And again, just as a key point, genome-wide association studies for binary outcomes will report the log odds ratio, and this needs to be exponentiated, which is just a function that we can use and calculate. So we exponentiate that to form the odds ratio. I've mentioned the p-value. I just want to make sure everyone understood what a p-value is. So the p-value is the probability of the event or of us getting that finding by chance. So given that there was no true association between a genetic variant and the outcome, what was the probability of finding this association by chance? So that was what the p-value shows. It's a probability of getting this outcome by chance. So in science, there's always this controversy about, you know, p is less than 0.05. So this means that we would accept a finding as being significant or unlikely to be by chance if the p-value was less than 5%. less than 0.05. So this means that any finding, for example, could be a false positive. So we could find that there's an association between a SNP and a trait, but this could be a true positive, but it could also be a false positive because, and it could have occurred by chance. Okay, and however, if the p-value is less than 5%, so that's like there's less than a 5% probability that's an accidental finding, we say that it's significant. Okay, so the threshold in science that we usually use is 5%. If the p-value is less than that, we say that it's unlikely that the finding was by chance, and therefore we accept that this is a likely significant finding. Okay. However, in genome-wide association studies, the threshold to consider a finding significant is much lower. So it's p is less than 5 × 10⁻⁸. And this is known as genome-wide significance. So whereas in most of science, it's if you get a p-value less than 0.05, we say it's significant. But in a genome-wide association study, the SNP association has to be significant. For it to be significant, the p-value has to be less than 5 × 10⁻⁸. So going back to the data from the MAGIC consortium, we see that all of these are non-significant because the p-value is greater than 5 × 10⁻⁸. However, on the last row, we see that this one is actually the p-value associated with this allele at that position, and this is the coding for the SNP. That is 5 × 10⁻�68. That is highly significant. So it's really unlikely to be a false positive. Okay. And why do we have this lower threshold for considering something to be significant in a genome-wide association study? That's because of this concept known as multiple testing. So I'm just going to explain this in briefly, but every time we make a test, there's a risk of having a false positive, and this is known as a type 1 error. Okay, there's always a risk that what we observe could be just by chance. However, if we're doing millions of tests, so because the data from a genome-wide association study, because we look at like I think something like 10⁶ genetic variants, there's millions of opportunities to have a false positive, and these increase at quite an alarming rate the number of tests that you do. So the risk of having, if we just do one test, the risk of having at least one false positive is 0.05, and that's seen as a kind of scientific convention. Okay. However, the risk, if we do 10, if we're testing, if we make 10 different tests, increases to almost 40%. And I put in a little bit about the maths here. So the probability of no false positive, if we were doing 2 tests, the probability of no false positive in the first comparison, that's because the probability of a false positive is 0.05. The probability of not having a false positive is 0.95. And then in the second test, you also have a probability of no false positive of 0.95. We then multiply that, and then to find the probability of at least one false positive, we subtract that away from 1, and that's how we get this figure. So this shows that there's almost like what, a 9%, 9.7% risk of having at least one false positive if we were looking at 2 different SNPs and testing whether they were significant or not. So as you can see, the type 1 error rate, so that's a risk of false positive, increases dramatically as we increase the number of tests. Because we're doing multiple testings for significance for all these different SNPs, we have to adjust what threshold we use. We reduce the threshold to make it more stringent and to make it more difficult for a SNP to be considered significant. And this is known as the Bonferroni correction for multiple testing. So in the case where we're making multiple tests of significance, so for all of these different SNPs, we're testing whether it's significant. Because of the risk of an increased false positive given the genetic variant, we have to adjust the threshold that we take for something to be considered significant. So usually our threshold, as I said, is 5%. If it's less than, the p-value is less than 0.05, we consider it significant. But to adjust for this, we divide the original significance threshold, which is known as alpha, by the number of tests that we make. And this is typically in a genome-wide association study is 1 by 10 to the 6th. And that gives us the output of the new threshold, so the Bonferroni corrected threshold, which is 5 by 10 to the 8 minus 8. So if the p-value is less than 5 by 10 to the minus 8, we can then say it's significant, and it's significant at genome-wide significance level. So genome-wide association studies are becoming increasingly more prevalent as there's more and more genetic data available. And these consortia, which are large groups, oftentimes publish summary level data for the genome-wide association studies. And that's similar to the type 2 diabetes data I showed previously. You can download these from the internet. They're quite large files. Some of them can have over 2 million rows, but they're publicly available. And these show how associations between genetic variants and different outcomes and traits. Essentially the likelihood of having a disease if it's a binary trait, or the association between a continuous trait and the genetic variant. So this is really quite a lot of information on the internet that we can use. And examples of common genome-wide association studies, for example, there's the cardiograms, which looks at coronary artery disease. There's this chronic kidney disease, CKD-GEN, GWAS, which looks at kidney disease. Psychiatric Genomic Consortium publish GWAS for conditions like schizophrenia. We see that there's lots of common genome-wide association studies that we can obtain summary data for. This is quite a good resource to see, well, what genome-wide association studies are available. So I'd recommend having a look through that, and you'd be surprised by the number of genome-wide association studies that have been done. Okay, I think it's time for a break now, because that's quite a lot of content. After the break, we'll be discussing Mendelian randomization, how we can set up an MR study, and some of the assumptions that we need to make for this type of study as well. But before the break, we had discussed a lot about the limitations of some research studies. So like we were saying with observational studies, there's the risk of confounding and reverse causality, and the issues with interventional studies, and that they're quite it can be quite expensive to set up. They can be quite time, and there's also always risks to humans when we make interventions, such as starting new drug therapies. So there's quite a lot of limitations in terms of both of these study designs. And when we look at current drug development pipelines, also it doesn't seem to be very efficient, because efficacy is oftentimes only tested late in drug development. So the initial is mostly exploratory in phase 1, and we look at pharmacokinetics, pharmacodynamics, and then it's only really towards the end of the drug development pipeline that we actually test whether it's effective versus either placebo or existing standard of care. And we see that actually that's, efficacy is one of the key reasons why a lot of drug therapies and a lot of new drugs fail in their development. So this study describes this quite nicely. So on the x-axis, we have different kind of classes of, or we have different drug, different disease states. So for example, in cardiovascular disease, we see the blue bars efficacy, and we see that led to maybe a 35% of the failures in cardiovascular drug development was associated with efficacy. This is higher for antineoplastics, so kind of drugs treating cancers. It's very high for respiratory, central nervous system drugs, musculoskeletal, GI, and systemic anti-infective agents as well. So you can see that efficacy is one of the key reasons why drugs fail in their development. And this is mostly tested in the end. So actually that could be one of the reasons why drug pipelines are quite ineffective processes. And this leads to increasing interest in having earlier assessment of whether a drug target could be associated with downstream benefits in terms of efficacy in humans. And this is where Mendelian randomization comes into play. So the key idea behind Mendelian randomization is that genetic variants, so these single nucleotide polymorphisms, for example, they're randomly assigned during meiosis, and this is according to Mendel's law of independent assortment. This says that the probability of having a gene is independent of the probability of having another gene. So actually, it's a completely random process. And this, in fact, mimics the randomization that occurs in a randomized controlled trial. So you can see that actually so if we study people according to their genetic variant that they have, that all the other confounding factors, so biological factors, social factors, are otherwise balanced if we study people according to their allocated genetic variants. So actually, we can use genetic variants as a tool to explore exposures and outcomes and account for some of these confounding factors. So it's a really key idea, is that we're using genetic variants as a proxy for an exposure and outcome, but because these genetic variants are randomly assigned, they're independent of confounding factors, and all other confounding factors will be balanced between these groups. So if we look at them according to the allocation of genetic variants, can say that it's unconfounded. And drawing out that point a little bit further, so essentially we can conceptualize Mendelian randomization as a biological randomized controlled trial. So on the left, we have the typical schematic for a randomized controlled trial. So again, we have the random allocation to either the treatment group or the control. And because it's random, we get the benefits of that confounding and being balanced between the groups. And then if we compare the outcomes for patients who receive the intervention versus the control, we can say that, well, the only difference between these groups was the treatment. Therefore, it's likely that this treatment is beneficial, or treatment is mediating that difference. So we can infer causality there. In Mendelian randomization, it's quite a similar process, actually. So instead of studying the effect of an intervention, we look at a genetic proxy that can act as exposure. So we can look at whether you have that genetic proxy or not. And then if we follow these people up over time to look at the outcomes, we can say that, well, actually that genetic proxy was causal and was a causal effect for that outcome change. The other thing to note is that because genes are assigned at meiosis, there actually is no potential for reverse causality here, because the outcome can only happen after the exposure, so when your genes were assigned at meiosis. So that's a key point as well. So actually, Mendelian randomization as a biological randomized controlled trial reduces the risk of confounding and also eliminates reverse causality. I sometimes think it's easier to think of these things using an example as well. So let's say a drug company wants to investigate whether iron therapy is associated with a reduction in the risk of cardiovascular disease. Ideally, this would be looked at using a randomized controlled trial, but we've run out of money. We don't have the time to study this in the form of a randomized controlled trial. We could approach this using observational studies. So we could look at people who took iron in the community and then follow them up over time to see whether they had different rates of cardiovascular disease in the form of something like a cohort study. However, there could be confounding factors. So people who take iron therapy might be more health conscious. They might also be prescribed something like a statin, which would also reduce your risk of cardiovascular disease. So teasing out the relationship between iron therapy and cardiovascular disease is, would be very difficult in an observational study because of confounding and reverse causality. However, we could do this using a Mendelian randomization approach. Okay, and I'll show you how that would work. So Mendelian randomization, again, a biological randomized controlled trial. So I'll go through this diagram here first. So instead of treating people with iron or control, in the case of a randomized controlled trial, instead we use genetic variants that mean that a person has higher systemic iron on average compared to a person who doesn't have that variant. Okay, so you would find these variants using Genomewide association study, or else we could find these variants using our biological knowledge, knowing that what variants are associated with high iron. And then we randomize people according to their allocation of these genes. So we look at if you have this SNP, is your outcome of cardiovascular disease different if you don't have that SNP? So again, we're looking at high iron, and we use a genetic proxy for that exposure. So we look at people who have a genetic propensity to a higher systemic iron level. And then we follow them up, and then we look at whether actually people who had that genetic proxy had different rates of cardiovascular disease. And in this way, we look at how the exposure can influence the outcome. So Mendelian randomization, you always need to think of two things. So you need to think of the exposure and the outcome. And we assess if people who have the exposure have different rates of outcome. And we use this with genetic proxies. So that's really the most powerful concept and that drives Mendelian randomization, is that we're using genetic proxies to represent exposure on an outcome. And the benefits of this, just to re-emphasize the point, is that because of genetic variants are randomly allocated, we're balancing confounding factors, and because they're assigned at meiosis before any of these outcomes can occur, there is no risk of reverse causality. So again, the formal definition here is that Mendelian randomization, or MOR, is the use of genetic variants to act as a proxy to study the effect of an exposure on an outcome. And we can visualize this using what's called direct acyclical graphs. So here we have the genetic proxy, or the single nucleotide polymorphism. Here we have the exposure, and here we have the outcome. And the association between the genetic proxy and the exposure is called the beta GX, and the association between the exposure and the outcome is beta XY. Now, what I don't have here is, on this graph, is the exposure between the genetic proxy and the outcome, and that would connect G to O, and that would be beta GO. And these beta refers to the regression coefficient from the Genomewide association study. Specifically then, if we are looking at the causal estimate, we're looking at, well, what is the association between the exposure and the outcome? This beta XY here is known as a causal MOR estimate, and that shows, well, the association between the exposure and the outcome as causal. And it's, if the assumptions of Mendelian randomization hold, that is, then that would be the causal Mendelian randomization estimate. Now, there's a number of assumptions that we need to make for a Mendelian randomization study. And the first assumption is that there's a strong association between the genetic proxy and the exposure, because ultimately the genetic proxy is what's driving the study. We need to make sure that that is a strong marker for the exposure. Okay, and this can be measured using several different metrics. So we use the R-squared value, and that shows the proportion of variability in the exposure that is explained by, I'm using this term, instrumental variable, and instrumental variable just refers to the genetic proxy. so the R-squared shows what proportion of the exposure can be explained by the variant. And if this is high, this means that, well, actually the variant is very strongly related to the exposure, and that's a key assumption for Mendelian randomization. The F statistic is another measure of the strength of the association, and by kind of convention, if it's greater than 10, we consider the association to be strong. So assumption number one of a Mendelian randomization study is that the genetic variant must be strongly associated with the exposure. Second assumption is that the genetic proxy is not associated with the confounding variables, so that this is independent of any confounding variable. Again, confounding variable being that it's a common cause of the exposure and the outcome. And then the final assumption is that the only pathway between the genetic variant and the outcome only occurs through the exposure. So the genetic variant cannot affect the outcome in any other way, only through the exposure. So let's think of an example of this. So if I was studying the effect of a genetic variant, if I was studying the effect of something like cholesterol on cardiovascular disease, I would use a variant that would mean that someone would have high cholesterol, and then I would look at their outcomes being cardiovascular disease. But this genetic variant can only affect cardiovascular disease through the cholesterol pathway. If this variant affected something like blood pressure, which can also mediate the risk for cardiovascular disease, then that would be an invalid genetic variant, and it would mean that they're actually the association between this genetic variant and the outcome is operating through an alternative pathway, and this is known as pleiotropic pathways. So we're talking about the concept of pleiotropy, meaning that there is alternative pathways for the association between the genetic variant and the outcome. But for Mendelian randomization, a key assumption is that actually the genetic variant can only associate with the outcome through the exposure of interest. So again, pleiotropy refers to the concept that something can have diverse effects. So for example, we can talk about pleiotropy in several different contexts. So statins have pleiotropic effects. This means that they can cause the lowering of lipids, but they can also have anti-inflammatory effects, for example. So we talk about diverse effects of drug actions. Similarly, genes can have pleiotropic effects, so they can influence several different phenotypes. So for example, let's take a hypothetical example that one of the genes that is involved in the synthesis of cholesterol could also be involved in the synthesis of, let's say, a different form of cholesterol. So the gene could be responsible for HDL and LDL, so good and bad cholesterol. So that's a pleiotropic effect, for example. But again, for Mendelian randomization, the assumption is that the genetic variant only associates with the outcome through the exposure with no other pleiotropic effect. There's two different types of pleiotropy, so vertical pleiotropy and horizontal pleiotropy. Vertical pleiotropy is actually what's happening in Mendelian randomization anyway. So the locus here, or the genetic variant, is associating with one trait and then it's associating with another trait. So this is all on the same causal pathway. There's no alternative pathway. It's vertical, happening along the same pathway. So for example, if I was looking at something like a genetic variant involved in the synthesis of cholesterol, and then further down the line, it was also associated with another, for example, was associated with another, I think that's a bad example, but basically if the genetic variant is associated with traits that are all on the same causal pathway, it's seen as vertical pleiotropy, and actually that's acceptable within a Mendelian randomization construct. However, horizontal pleiotropy, which is when there's alternative paths, so for example, you could have a genetic variant involved in the synthesis of HDL, but also involved in something like triglycerides or LDL. These are alternative pathways, and these would be invalid. So we always have to consider pleiotropy as a potential issue at in Mendelian randomization, and there's ways that we can go about exploring this in our analysis. But for a valid Mendelian randomization study, the genetic variant only associates with the outcome through the exposure with no horizontal pleiotropy. And then there's a couple more terms that I'd like to explain. So oftentimes in studies, you might see the concept of two-sample Mendelian randomization. And this refers to the fact that the exposure and the outcome come from two different genome-wide association studies with no overlapping populations. And the benefit of this is that you get increased statistical power because there's a lot more individuals being included. So you have a cohort who are looked at for the exposure and a different cohort who are looked for the outcome. So we're actually using a lot more sample size in the Mendelian randomization study who are contributing to the association between the genetic variant and the exposure and the genetic variant and outcome. So how do we calculate the beta XY, so the association between the exposure and the outcome, or the M-OR causal estimate? So we do this using the Wald ratio. And this is simply a division. So what we do is we get the associations, we get the beta regression estimate from the genome-wide association for the exposure. So we look at what is the association between the genetic variant and the exposure. And then we look at the genetic association between the variant and the outcome. And once we have these two green associations, we can then do some simple rearranging to find, well, what is the association between the exposure and the outcome. So we know that the beta GY, so that's the association between the variant and the outcome, is equal to the association between the variant and the exposure multiplied by the relationship between the exposure and the outcome. Then we can rearrange that to say that the relationship between the exposure and the outcome, so beta XY, or the M-OR causal estimate, is equal to beta GY divided by beta GX. So that's the outcome over the exposure. So the M-OR causal estimate is equal to the beta for the association between the variant and the outcome divided by the association between the genetic variant and the exposure. That calculates this association, that is the causal estimate for the exposure on the outcome. And as we saw in the genome-wide association study, that there's always a margin of error associated with all of these regression and beta coefficients. And we need to take this into account when we do a division, because these errors propagate as we manipulate the measurements. So for example, there is an associated error, standard error, with the beta GY that we can get from the genome-wide association study for the outcome. And there's also a standard error associated with the GWAS for the exposure data. Because we're dividing these, the errors will also propagate along with the manipulation. And this is done using a formula where we divide the standard error for the beta GY by beta GX. So this is just a way that we can calculate the standard error for the M-OR causal estimate. So that is this association using the standard error and the beta coefficients from the other data as per that formula. So again, this is just showing that how we get the standard error for the M-OR causal estimate. So putting this all together, we're going to do a simple Mendelian randomization study. And essentially there's 5 key steps that we need to take for any Mendelian randomization study. So we need to get the genetic data from genome-wide association studies for the exposure and the outcome. And this gives us, as I said, the the beta coefficients, so the strength of the association between the genetic variant and the exposure, and the genetic variant and the outcome. Then we harmonize the genetic data to make sure that all the alleles match and that the data is compatible. And then we use the Wald ratio to calculate the MR causal estimate, and then we calculate the adjusted standard error for the Mendelian randomization causal estimate. So we're now going to do that for Mendelian randomization study. So I'm taking some of this data from a paper that was published by Dipender Gill in 2017, which looked at the effect of iron status on risk of coronary artery disease. So this really addresses the question of whether iron could prevent coronary artery disease. So I've kind of taken some of the data from that study as an example. I appreciate this is a simplification. I'd recommend that you go into the paper in more detail to understand more about the methodology and the detail of the study. But just as a simple way to understand the calculations, we're going to use it as an example. So let's say you are the pharmaceutical company who didn't have the funds to test whether an iron tablet would help with the prevention of coronary artery disease. We're going to do this using a Mendelian randomization approach instead. So first of all, we're going to get the outcomes for the exposure. So here we've found 3 genetic variants that are associated, strongly associated with systemic iron level. Okay, the p-value here are all less than 5 × 10⁻⁸, so they're significant. The SNP, this is their ID, individual to each genetic variant. If you have this allele at that SNP, at a certain genetic position of the genetic code, there's an increase of 0.33 in the systemic iron level. Then this has a reduction, and the third one also has a reduction in iron. So these are at a population level. People who've had these genetic variants have different levels of systemic iron on average. Okay, and we see the F statistic, which is a measure of association, and that shows that these are all quite strong, and the R squared gives us an idea of what percentage of the variability in iron could be explained by this allele. We see that this is quite low, but that's not surprising because single nucleotide polymorphisms tend to describe only a small amount of the actual difference in phenotype or the physical trait. And then the effect allele frequency just shows how frequent they are in the population. So this allele is 70% seen in 70% of people. So we find that these variants are strongly associated with systemic iron level at a genome-wide significance level, and these are what we're called instrumental variables, but we think of them as genetic proxies to mimic iron exposure. Also, it's important to note that these exposures are lifelong. Okay, so it's not, whereas a drug, for example, you take it and the effect can be quite short. These people on average would have higher systemic iron over their lifetime. So it's kind of a cumulative effect. And then step 2 is we're going to get the association, the genome-wide association data for the outcome. And this shows the association between the same variants and coronary artery disease, and this was obtained from summary level statistics for genome-wide association studies. And again, we find beta GWAS, so that is the association between G and Y, between a genetic variant and the outcome. And then this is the standard error and the p-value. It's important to note that the effect allele for the outcome and the exposure has to be matched. So we have to harmonize them. If this is different, we have to adjust the signs of the association to match the alleles. We'll go into this in a little more detail when we start doing more Mendelian randomization studies, and there's computer packages that can help us do this, but it's important to know that the effect allele here has to be the same as the effect allele in the exposure GWAS. So we have on the first SNP, ORS1800562, the effect allele was A. And in this, for the outcome data, same SNP, the effect allele is A. So they match, so they have to be harmonized. And then we merge them together. So this is the merged harmonized data showing beta GX and beta GY. Beta GX, again, is the association between the genetic variant and the exposure here being iron, systemic iron levels, and beta GY, where the exposure, which is the association between the genetic variant and the outcome. And these, again, are harmonized. And now we're going to calculate the Wald ratio. And as I described before, the Wald ratio is simply a division of the association with the outcome divided by the association with the exposure. And if we get our calculators out, this is the result. And because this is the unit, because coronary artery disease is a binary trait, so either have coronary artery disease or you don't, this outcome is the log odds of coronary artery disease per increase in the effect allele. So this is really important to note, that the outcome unit is the log odds of coronary artery disease per unit increase in the effect allele. And then we convert that to the odds ratio, as I've said, using the exponential function. And that gives us the odds ratio for coronary artery disease for every one of these effect alleles that a person has. So for example, here we can see that there's an 8%, roughly about an 8% reduction in the odds of coronary artery disease for every copy of this SNP. And then we calculate the standard error using this formula. It's important to note that we only exponentiate the outcome at the end, so once we have taken into account the standard error. And this gives us the confidence interval. So if the confidence interval crosses unity, we then say that it's a non-significant finding. However, for example, here that the 95% confidence interval is always less than 1. We can say that that's a significant finding. Okay, so this suggests that there is a reduction in the odds of coronary artery disease, and that we're 95% probability that the true odds ratio lies between 0.79 and 0.11. So for this SNP, this first SNP, we can say that if someone has that SNP, they are less likely to have coronary artery disease, and that would be mediated by reduction by the changes in systemic iron exposure. We can display the results of Mendelian randomization study using a forest plot. It's quite a common way that they're displayed in MOR studies. So here we have on the x-axis, we have the odds ratio, 1 again being unity, that there's no effect of the SNP on coronary artery disease, or in this point, serum iron. So for the first variant, ORS1800562, we see that there is a reduction in the odds ratio of coronary artery disease with the effect on serum iron, and that this is below unity with the 95% confidence interval below 1. And this means that that is a significant finding, that there's a reduction in the odds ratio of coronary artery disease associated with serum iron. However, for other SNPs, this is not the case. This could be quite a common finding, that some SNPs would be associated and others wouldn't be associated. And for that reason, we tend to pool these results together using a term called inverse variance weighting. And that's to make sure that that we're pooling many different results together to get a more accurate representation of the effect of iron on coronary artery disease. However, that's for another session. At the moment, I just want to emphasize the actual calculation for how to get the M-ORA causal estimate for a single variant. And then at another stage, we'll come back to how we actually merge the results of many different SNPs together to get a more accurate representation of the true effect of iron on coronary artery disease. So we covered quite a lot today, but in further sessions, we'll discuss how we're using multiple genetic variants and pooling these results, and that's using the inverse variance weighting and other pooling estimates. And we're going to talk about sensitivity analysis, because we need to make sure that these findings are robust, and there's several different techniques that we can do to assess that. We also need to look for pleiotropy, and that's where there's alternative pathways. We need to make sure that the variants are valid in, representations for the exposure and the outcome. So today we've done the calculation by hand. However, there are coding packages and OR that will assist with calculations. It could be quite useful, but also it's important to note that we need to understand the actual mechanics and the basic mathematical concepts that are underpinning Mendelian randomization to, I guess, to fully understand our analysis. But what we did cover today is that we covered study designs, particularly randomized controlled trials and observational studies. We discussed a lot about confounding and reverse causality and why it's important to consider these within observational studies. Then we had a discussion on genome-wide association studies, but ultimately the main emphasis today was Mendelian randomization, how we can use genetic data to proxy the effect of an exposure on an outcome. And this overcomes a lot of the issues with traditional observational studies such as confounding and reverse causality, and how actually it mimics the the benefits of a randomized controlled trial. Then we've done a simple walkthrough of a Mendelian randomization analysis. However, there's a lot more to learn around the sensitivity analysis and further more in-depth statistical techniques that we can use to make sure the Mendelian randomization is valid. So that's all that I wanted to cover today. Thank you very much for your attention.